Okay, so people are probably going to start to roll in a little bit, and that's all right if we kind of get started. This is the UWBC Biotech Talk, virtual biotech talk for May. This will be the last biotech talk we have probably until the start of the semester in September, when we're deciding if we're going to keep with a hybrid or what kind of technique or method we are going to use for our biotech talks in the fall. But keep on the website and we'll have updates coming up this summer. So I am actually going to pass it off to Bridget right now, who is going to introduce our speaker.
Hi, I am Bridget Liddell, and I'm a special projects researcher with the DNA sequencing facility here. And so one of the things I get to do is start validating new services and new techniques that we want to start offering everyone to add on to their projects and research. And something we've been working on is Hi-C with the Arima Genomics kit. And we've gone through a few different tissue types, and we've had a lot of success with the projects we've had so far. And so we wanted to bring the team in and have them talk about the kit and about the process, and hopefully, have people have a new project they can add to their research or new technique. And we're gonna have Allyson take questions in the chat. She's head of technical support, so if you have any questions, you can ask them in the chat, and she'll get back to you. And we'll also do Q&A at the end as well. So pass it off to Brenda.
All right. Thank you very much, Bridget. It's very nice to meet you all. Thank you for your time today and for having us. As Bridget mentioned, my name is Brenda Trevino, and I'm with Arima Genomics. I'm a strategic account executive. Also on the call today is Allyson Whittaker. She's our head of technical support, and she's an expert in all things Hi-C. So we're very, very happy to have her today. So first, I'd like to go over the outline of what we'll be talking about. Just really quickly, we'll go over what essentially is 3D genomics and how it can apply to you, an introduction to Arima technology, and specifically talking about using Hi-C for genome assembly, epigenetics, and disease mechanisms. And then lastly, our latest and most exciting launch has been in the area of FFPE samples. So we're now able to analyze and process FFPE samples that have been archived, even up to 12 years. So I'll be talking a little bit about that and the types of data that you can obtain from those types of samples. So Arima Genomics, we're based in San Diego, California, and we focus on helping science, working for scientists and helping scientists advance their research. So our leaders were founded, our CEO is Sid Siddharaj, who is from Bing Ren's lab. And he founded the company in 2015, starting with the Hi-C technology. We're currently a 35-person team and still growing. And we have under our belt, thanks to the support of many customers such as you, 250 customer publications. And so that's something that in the short time that we've been around, we were started in 2015, and lately we have so far accumulated approximately 250 different publications using our technology, and so that's an accomplishment that we're very proud of. So we offer technology for using Hi-C in the form of kits and reagents, and also services and support. So next, I'd like to go over what essentially is 3D genomics. So looking at the 2D or linear genomics, there's a lot of different assays that I'm sure you're very familiar with, such as DNA sequencing, RNA sequencing, whole exome sequencing. But it really leaves out the context of looking at the structure of what's going on within that linear space. And when you do look at the the additional 3D context, you also get insight into the 3D structure. The structure of that particular chromosome, and therefore any structural variants that might be involved. You also see the regulation of different genes and the organization from such things as compartments, tabs, and loops. So without all of this context, we really do miss a lot of what's hidden under the surface, and are really just looking at the tip of the iceberg when we look at the 2D genome. Looking a little bit more closely at the 2D genome, we can see here the organizational architecture. And what you can see is a top-down view of the nucleus. Each one of these distinct colors is a chromosome territory. What we found is that chromosomes are organized into distinct territories, mostly self-interacting, versus a mixture sort of similar to a lump of spaghetti in a bowl, and randomly associating with many other chromosomes. These territories are organized differently in that they're highly condensed and self-interacting. So drilling down further into the chromosome territory, what we have here are the compartments. And these compartments really show that you can identify any A-B structures. So they're organized into A and B compartments, with A and B being A being the active regions and B being the inactive regions of a particular chromosome. And also drilling down more closely into the compartments, we begin to see structures such as tabs, or topologically associated domains, in which we start to see loop structures form. And in this diagram here, you can see the boundaries between the loop formations marked by CTCF. And then looking more closely within these topologically associated domains, we start to see the individual loop structures here that are bound by cohesin. Oh, sorry about that. And what this essentially shows is that there's a number of insights that you can make into the biological relevance, and therefore the pathological implications of disease. Looking at loops specifically, if we were to take a look at data showing all of the looping interactions for a particular region, we can infer communication between promoters and enhancers. You can start to see, for instance, coordinated gene expression, and therefore also infer the pathological implications, such as what fusion transcripts are being associated with that particular oncogene, or any types of long-range effects with SNVs. And so what this really highlights is that Hi-C is a very important tool for human health, and it enables researchers to be able to detect and discover pathological implications and advance their research. So before we dive into the applications and examples, what I'd like to do is just review the workflow from a very high level. Looking at the Hi-C workflow, we start with a wide variety of different types of cells and tissues, and that varies from plants, insects, FFPE tissue, frozen tissue, cell lines, blood. All of these differ in how we prep the DNA. And then from there, the protocol is the same for all of these types of samples. So it's a very robust technique in that once you do use a particular sample type, let's say a plant sample, and then you would like to proceed with, say, an FFPE type sample, the only difference is going to be that initial starting sample prep. And then the rest of the workflow is going to be essentially the same. And so after the sample prep, what you do is you perform a restriction enzyme digest, and then the ends are labeled. I'm sorry, the 5' ends are then labeled with biotin and ligated. The DNA is then purified, and then you go into fragmentation and enrichment with biotin. So that leaves your proximally ligated material. You can see these are chimeric. We have one purple and one blue. And so you have your proximally ligated DNA material. What's really nice about this workflow is that it also works with a single tube chemistry, which minimizes sample loss should you be working with low input amounts. The protocol is also very fast in that there's only 1 hour of hands-on time, and the protocol itself is about 6 hours. And there's no special instrumentation that's required, just your typical lab equipment like a thermal cycler, a bioanalyzer, qubit, or tape station. So once the Hi-C reactions have been done, the process is very straightforward, similar to other NGS methods, in that you'll go into a library prep, your sequencing, and then your data analysis. So this makes it very easy for your core provider to be able to process your samples and to be able to work them into their current workflow. One thing about the assay is that it is more sensitive than whole genome sequencing due to the proximity linked reads, and therefore the efficiency of the protocol and use of the multiple restriction enzymes gives you more cut sites and data points, and thereby reducing signal and background noise in your data. So while you do get deep sequencing reads, the reads are tailored to that specific region of interest. And so you have the majority of your data is tailored to that one specific long-range cis read. Okay, so why explore the 3D genome? Well, there's a couple of different areas that I'd like to talk to you about today. One is genome assembly, the other is focused on epigenetics, and the third is human health. First, looking at genome assembly, you can use Hi-C for building fully phased genome assemblies that enhance the ability to advance precision medicine. You can also use it for correcting assembly errors such as scaffolding contigs and providing chromosome spanning contiguity to any assembly. And also, I'd like to note that Arima is a trusted partner with the Vertebrate Genome Project, which has been used to help create reference quality assemblies of more than 260 species. And you may be familiar with the paper that was recently published on the human genome by the T2T Consortium in 2022. Arima is proud to have been a partner on this exciting project. And although the genome was mapped, the reference human genome was mapped in 2000, approximately 5% was still missing or incorrect. And the main challenge was that these regions were highly polymorphic and that they have many repetitive regions that span several megabases. So the researchers used Hi-C reads in conjunction with long and short read technologies. And so what we're showing here is data from the T2T project that shows a typical heat map from Hi-C. Along the x-axis are your chromosomes, so chromosome 1 through 22, and along the y-axis also chromosomes 1 through 22. The deep red color is the number of reads that were assigned to that particular interaction. So as the color gets more intense, the more reads support that particular linkage. And since the the chromosomes tend to be highly self-interacting. What we find is that a nice diagonal line is created. For instance, if we were to have a linkage at chromosome 7 and also at chromosome 7 here, we would see a point here just supporting that diagonal line that we see, and proving that most of the data is self-interacting. And this data here shows that that the end point of the data that was fed into the T2T consortium algorithm was highly contiguous, very well defined, and as a result, the researchers were able to create, or not create, but add an additional 200 megabases of sequence to the reference genome. Here's another example of using Hi-C for genome assembly, in which chromosome scaled phased assembly was done without parent genomes. And what was done in this case is Hi-C reads were used along with PacBio reads to create your phased contigs. What was found was that by utilizing Hi-C reads, the researchers were able to use their new algorithm, the Dipasm algorithm from Shilpa Garg, to be able to have scaled assemblies, chromosome scaled phased assemblies within 1 day. So they were able to utilize Arima's Hi-C technology along with long-range reads in order to create these phased contigs that they previously they did not have, since they did not have parent genomes. So our approach for data analysis of genome assembly is that we are an open source supporter of any types of algorithms or data analysis tools. We work with you on the the installation of the pipeline and the implementation. We also provide the restriction enzyme cut site information, and we provide data quality interpretation. For instance, if you were to send us your shallow sequencing data and your FASTA file from your initial draft assembly, we'll actually subsample that data and do a QC analysis and interpretation for you at no extra cost. We just really try to be there for you throughout the way, also helping with the development of any open source algorithm tools. For instance, we worked with the development of the SALSA2 algorithm from Adam Phillippi's lab, Instagrail from Romain Causel's lab, and as I mentioned a moment ago, the Dipasm Pipeline from Shilpa Garg. What this is showing here in this cartoon is just the typical workflow of the data analysis pipeline, showing Hi-C reads used in combination with your contigs or your scaffolds, and into the SALSA2 program. With each iteration, a file is created until no changes or no additions are made to that particular file. And therefore, you have a final result of a highly contiguous assembly. And so what that looks like here is that we have several heat maps. One is our starting iteration showing nanopore contiguous, or data from nanopore. The contigs were then placed along this diagonal, sorry about that, this diagonal line here. And what you can see is that the signal is not that bright, and that a lot of the contigs still remain in the interchromosomal space. So there's a lot of assemblies that are that still need to be be properly aligned. For the third iteration, you can see a more intense signal where a number of these reads now been added to the appropriate contig and are there aligned here in the third iteration, the contigs are more defined, and by the sixth iteration, the contigs are even more highly defined, with less of the contigs in the intrachromosomal space and more of them in the interchromosomal space. So essentially what you see is that the contigs are, since they're likely derived from the same chromosome, each iteration stitches them together and you start to see that clear definition along the diagonal, which is what you're looking for. So why explore the 3D genome? There are lots of ways to use Hi-C. One is to use it for epigenetics and yielding new insights into gene regulatory environments, such as capturing epigenetic mechanisms that regulate transcription. There's also the easy use of combining assays such as Hi-ChIP and Hi-C with capture Hi-C. So you can get insights into promoter enhancer regions or specific genes using a capture-based approach. And we also provide a multi-omics wet lab and dry lab workflow. With the dry lab workflow, what we provide is the data analysis workflow for your tools that are needed to analyze your data. So the Hi-ChIP workflow is very similar to Hi-C in that you can start with any type of input material as previously mentioned, and then it goes into the Hi-C workflow, also as previously mentioned. But then what you also can do is you can enrich that sample for a specific target or protein of interest, such as H3K27 or CTCF. And then you will have your proximally ligated DNA that's specific to that particular target of interest. At that point, you'll go into the library amplification and then your deep sequencing, very similar to Hi-C. And with capture, our capture-based approach is also very similar in that it's also a bolt-on type of assay in which you start with your Hi-C, similar to the previous method that we discussed. And then you perform an enrichment using capture probes after you amplify your library. So you perform a hybridization with your capture probes. You PCR amplify your library, purify your samples, and then go on to your deep sequencing step. So I just wanted to touch on the capture Hi-C while we're here talking about Hi-ChIP. Looking at the Hi-ChIP workflow, Arima has a sort of a one-stop type of analyzing the data in that we use Feather, Max2, and MAPS. What this enables is with Feather, you can create a chip enrichment metaplot to ensure you have enriched with that chip antibody that has occurred properly. You can also then use ARC plots, load your data into the Wash U epigenome browser. You can have your loop calls paired with ARC plots for visually assessing the chip enrichment. And then there's also the QC data. With each Hi-C kit with Arima, what we have is several QC steps along the way. And the QC steps are done to ensure that the reactions have occurred thus far to a certain criteria. level. And with those benchmarking guidelines, we're then able to predict the success of the reactions going forward. And so here, what this is showing is that you're able to utilize your data pipeline here and predict the success and assess the quality of your high chip data. Here's one example. It's a very interesting case study in which the high chip assay was used by Wilson Tan and his colleagues at the Cardiovascular Research Institute in Singapore, in which the researchers studied diseased and non-diseased human hearts in order to capture non-coding genetic variants and in enhancer elements associated with heart disease and heart failure phenotypes using the H3K27 acetyl ChIP-seq high chip kit. And they were able to identify disease-associated genes that were linked to those enhancers, showing the first the first instance of use on with high chip on human hearts. Looking at the data in a little bit of a more specific way, what they showed here was they were able to look at a specific SNP, the SNP RS12143842, and which is approximately 5 kilobases from the NOS1AP promoter. This allele is associated with increased enhancer peak height and increased expression of the NOS1AP gene and the distal C1orf226 gene, but not with the OLFML2B gene, which is in a neighboring CAD, which you can see here. And that was surprising. So what you see here in the black circle is the chromatin loop that associated the connecting to distal loci, the NOS1AP and C1orf226. And this C1orf226 is located 500 kb downstream. So by identifying this particular SNP, the researchers were able to gain a target for therapeutic development. Also looking at why explore the 3D genome, we can take a more translational look at epigenetics and look at how we support novel insights into human health. For instance, you can use the data from Genome Assembly, from Epigenetics, and then further drill down into gathering insights for gene regulation and being able to identify structural variants, better understand disease mechanisms for transcriptional regulation. And you can also develop prognostic markers, new therapeutic targets, or even assess therapeutic responses to treatment in patients. So what we have here is just a quick slide looking at the breadth of how 3D genomics has been used in various disease research applications, from cancer to immunology to stem cell biology. There are several different types of areas of focus that can be used for determining 3D genomic interactions in your research project. This is a very interesting study. This was done by the Afontis and Sirigos labs at NYU. And this is all published data that you can look at in greater detail. And what you see here is a healthy population with a MYC oncogene here, an enhancer about 2 kb away. When you're looking at the linear genome, this is all the information that we have. However, when you look at the 3D genome, we can see that there are 2 loops that prevent the MYC oncogene and this this enhancer from interacting. And in TALL diseased samples from patients, we see the same, basically a very similar expression with your MYC oncogene. Now we know that there's transcription factor binding, but we don't see that there's, now that you have some misfolding going on, which is able to be seen only with 3D, with a 3D genomic approach like Hi-C, and be able to see that now without this, second loop, a transcription factor is able to bind to this enhancer, drive MYC expression, and now you have your MYC expression increased. So what this did was this provided an insight into what researchers could use as a target for drug treatment. And that's exactly what they did. So for patients who received a treatment or therapy, drug therapy, the function was restored. And once again, there was no interaction here between that transcription factor and enhancer, because now we have the folding pattern that has returned to normal. So what I'd like to do now is talk a little bit about what we're doing in the span of using FFPE samples to detect gene fusion events in cancer and how it affects human health. In a recent webinar by Dr. Matthias Snerdl, he was able to show several case studies that identified novel fusions that were missed by other assays, also detected clinically relevant fusions that were missed by DNA and RNA-seq and DNA methylation assays, and uncover fusions in FFPE specimens with degraded DNA, such as DNA that was approximately 12 years old. And looking at why 3D genomics is so powerful for cancer research, well, we know that DNA sequence is very important to being able to provide a foundation for being able to discover gene regulation and identify things like structural variants. So you can see here, by being able to pair all three together, that you can see a lot of insights into cancer. In fact, more than 95% of cancers have one or more somatic structural variants. At least 30% of cancers have a known pathogenic structural variant that's used in diagnosis or patient stratification. And structural variants can impact the regulation of cancer genes through 3D interaction. And so we feel that detection and characterization and understanding of structural variants requires an integrative approach, and is therefore a very important tool in the understanding of structural variants in cancer. So here's some data looking at chromosomes 2 and 5, and which once again we see here what we would expect to see from a normal karyotype. But you could ask yourself, what if there was a reciprocal translocation at chromosomes 2 and 5? What would that pattern look like on a heat map? And in fact, what we would start to see are these bow tie type patterns here, where we would have at chromosome 5 and chromosome 2 this particular appearance, and also here at chromosome 2 and 5. So you can see that this is a quick visual representation for showing that there is something that's going on out in the interchromosomal space and indicative of gene rearrangements. So looking at a normal karyotype, this particular karyotype is of a ROS1S LC34A2 fusion. You can see that there's a lot of off-diagonal 3D genome linkages in the abnormal karyotype compared to the normal karyotype. And there's a lot of high strength linkages, which would be very evident, this would be evidence for structural variants. So you can see how deep red these signals are. And that just shows that there's a large number of reads that support that particular interaction, showing that, again, that the evidence for that particular structural variant is quite high. Looking at data from FFPE samples and our partnership with Children's Mercy Kansas, Scripps, and MD Anderson, we had 24 FFPE samples, 19 tumor types, approximately anywhere from 1 to 12 years of archived time. And the amount of DNA that was restored or recovered was about 300 nanograms, although this spread was less than 10 up to over 1 microgram of DNA. And so looking more closely at this particular benchmarking study that we did with these institutions, we were able to detect 12 different tumor types, using 12 different tumor types. These types of fusions, you can see that the majority of them are EML4 ALK gene fusions, but there's also a number of different gene fusions that were detected. Also detected by orthogonal assays. Some of them had been also identified, just showing the concordance between our data and the orthogonal assay, and then the type of orthogonal assay that was performed. So this is very exciting for us because we were able to take various amounts of FFPE data, various amounts of FFPE DNA, and archived at up to 12 years, and generate the same results as the orthogonal assay, or even better in many cases. So what I'd like to do now is just highlight that we are offering a cancer grant application. The deadline is tomorrow. So if you are working on a cancer research project and you would like to receive a free Hi-C kit of your choice, it can be a genome-wide Hi-C kit, a HiChip kit, or a promoter capture, or even a capture-based Hi-C kit that is using probes that are custom to your specific project. You can win any of those types of kits. Just fill out this application on our website for the Cancer Research Grant, and the winners will be announced very soon. So that is something that's a very exciting opportunity. So to conclude, there is a lot more to the genome than just the sequence. 3D genomics approaches can provide novel insights for disease researchers, and we're so excited to have those researchers here today. Once again, just to highlight the importance of not just seeing the tip of the iceberg, but to be able to take the 2D sequence information that you have and being able to add a 3D context to it and really get the full picture of what's going on. So with that, I will open the floor up to any questions that you have. I know Allyson has been available for any questions as the talk has been going on. And thank you very much.
Feel free to unmute yourself and ask the question if you'd like, or you can put it in the chat as well.
So we have one question in the chat, which is what are the input requirements for genome-wide Hi-C, and also the depth of sequencing needed. So I mean, it depends on the input. We have different input requirements for cells versus animal tissues versus plants. So for plants, it's about 2 grams of plant tissue. For animal tissues, it's about 50 milligrams to 200 milligrams of tissue, although we do have low input. Arima excels at low input, so we do have protocols for a low input as well. And for cells, it's about, you can do Hi-C on about 1 million cells, but once again, we have low input protocols that can get you as low as, we've had success with as low as 50,000 cells as input. And so typically, so for something like human, the depth of sequence required for genome-wide Hi-C is about 600 million paired end reads, but it will scale depending on your genome size, right? Are there any additional questions? And remember too, that as you can see at the bottom of the slide, you have both Brenda and my email addresses, plus of course the organizers of the talks, if you have any additional questions about the Arima technology.
And this talk was also recorded, and it'll go up on the UWBC website probably within the next week, actually on our YouTube channel. So you can share it as well. All right, well, if there are no additional questions, thank you very much, everyone, for joining us. And as I said, if you would like a recording of this, it'll be up on the UWBC website within the next week. So thanks, everyone, for joining us actually for all of the biotech talks this year, and look out for talks in the fall. Thanks, everyone.
Thank you. Bye-bye.